Good afternoon, good morning, or good evening, depending on where you are. I'd like to welcome you to the Margaret C. Etter Early Career Award presentation. This award, I think, is probably the most important of the ACA because it recognizes the careers of promising young scientists when they most need the backing of their peers, which is in the early stages of their independent career. The award is established to honor the memory of Professor Margaret C. Etter, who was a major contributor in the field of solid-state organic chemistry. I personally never had the chance to meet Peggy as she was known to her friends, as I joined the ACA only a few years before her tragic loss. Peggy's work emphasized the use of hydrogen bonds and co-crystals, and despite the very few years she was given, resulted in a set of rules known as graph sets to describe hydrogen bonds in a way that revealed similarities between structures without being too tied up in the crystallographic details. She had considerable experience teaching at an undergraduate institution, but worked both in industrial and academic settings. And that gave her an unusually broad perspective from which to mentor students and to support and encourage colleagues. She had a great love for people, for science, and especially for people who do science. She was also much loved by her fellow ACA leaders, who established this annual award a decade after her death in 2002. The award consists of a prize of $1,000, a plaque, and the opportunity to speak here at the annual meeting of the American Crystallographic Association. Scientists involved in crystallographic research are eligible for this award. At the time when nominations are expected, nominees must be no more than 12 years beyond their PhD degree, which can include career breaks, and must have begun their first independent position within 10 years of nomination. Nominees who are in tenure-track positions must not have received tenure at the time they're nominated. Let's see. Here's a list of the previous Etter Award winners. You'll notice quite a few names that I suspect many of you will recognize. Many have gone on to be quite active in the ACA. The winner today is Nozomi Ando. Nozomi did her bachelor's work and her postdoc at MIT with a PhD from Cornell. She started her academic career at Princeton University and then moved back to Cornell University. Nozomi has received an NSF Career Award as well as 3 NIH awards. Nozomi has been the director of the Ariche School of Crystallography. I believe that's coming actually in 2 years, and served as one of the co-chairs of the this particular American Crystallographic Association meeting. She has been a featured speaker, either or a plenary speaker in quite a few conferences. During the preparations for this meeting, Nozomi shared one of her preprints, which I found clear and compelling despite my lack of experience in her field. She and her group study how enzymes work using a combination of biophysical and biochemical techniques. Their main focus is studying proteins in motion, and she's best known for X-ray scattering approaches to studying both static and dynamic conformational disorder. I'd like to once again thank Nozomi for her work in co-organizing this meeting, well, actually twice, since it was originally planned to be in person and now virtual, and even for making the background to these slides. Nozomi, I'm very much looking forward to hearing your talk. And congratulations.
Okay, hopefully everybody can hear me. First of all, thank you so much, Brian, for that kind introduction, and Kristen as well. I'm going to do my best to do this live. However, if we have technical problems, it will be okay. Okay, you can always email me. We are doing our best. So first of all, thank you so much to the ACA for this Etter Prize. It's a great honor, and it means a lot to me, especially coming from this community. And I have many people I want to thank, but I especially want to thank this group shown here in this photo. This is my lab. The students and postdocs are really what make my research program what it is. So I'm going to tell you about this sort of central idea that we in my lab like to study, which is protein allostery. So protein allostery is often described as action at a distance. And this is the concept that part of the protein can communicate with another distant part of the protein. And I like to think of this as a special property of macromolecules that connects molecular motion to action. And this is what enables proteins to be able to not only regulate its function, but also perform its function in the correct sequence. And so why do we study allostery in my lab? It's because I think it provides this unique window into understanding the fundamental relationship between the primary sequence of a protein, its three-dimensional structure, its dynamics, and ultimately its function. And to me, that is the ultimate goal of structural biology. In my lab, we're known for using several techniques. I think we're probably best known for using scattering, but I like to think that we like to solve a certain class of problems, and that in the most general sense involves extracting information from large data sets. For example, you'll hear a little bit about factorization of scattering data. In this talk, I'm going to tell you about two stories about two different aspects of protein allostery that we're studying and our approach to studying those problems. So the first story is about the evolution of allostery. And this is the work primarily of Will Thomas, graduate student. Many of you attended his talk yesterday. He's also session chair at this meeting. And because it's a special occasion, I also want to show the photos of all of the co-authors from my lab who contributed to this work in these papers below. Okay, so why are we interested in studying the evolution of allostery? And for me, it's because it gives us a way to relate protein sequence to protein allostery. And in my lab, we do this by studying a family of ancient enzymes that are essential for all DNA-based life. So I think many of you have heard of these macromolecules. These are very important for biology. And I think many of you may also know that RNA is a polymer. It's made out of ribonucleotides. DNA is also a polymer. It's made of deoxyribonucleotides. And some of you may know, I think probably most of you don't, but there's a protein that's responsible for converting ribonucleotides to deoxyribonucleotides. And it's called ribonucleotide reductase, or RNR. I know it's a mouthful. The important thing is it uses radical-based chemistry, and this enzyme is really essential for making DNA in nature. And because it's so essential, you can find RNRs in every organism except for maybe a few parasites. And as a result, we have a lot of sequence data on the RNR family. So this is a very simplified phylogenetic tree. And I've colored it in a way to focus on the class 1 RNRs. So here are the class 2 RNRs, which are also very interesting, but not the subject of this talk, is rooting this class 1 RNR tree. And this is the subset of the RNR family that's most recently evolved. And we have many sort of reasons to believe why it's the most recently evolved. One reason is that it includes all of the eukaryotic sequences. And so, for example, we are here in this family tree. So what is interesting about the class 1 RNRs? I'll tell you about that. I told you already that the reaction that this enzyme catalyzes is this conversion. And it's really remarkable because it's able to take 4 substrates and convert it into 4 different products. In the case of the class 1 RNRs, the substrates are ribonucleoside diphosphates, so 2 phosphate groups. And because this reaction is so essential for central metabolism, it's really tightly regulated with multiple forms of allostery. And the type of allostery I'm going to tell you about today is called activity regulation. And the way RNRs have figured out how to do this is really clever. It senses the balance in the cell of ATP versus DATP. So ATP is a substrate derivative. It has 1 more phosphate than ADP. And it tells the enzyme that the balance of this nucleotide pool is high, so that move forward with the reaction. And conversely, DATP, which is a product derivative, tells the enzyme to stop. It's a feedback inhibitor. Now, to understand how activity regulation normally works in the RNRs, we need to talk about the structure a little bit. So the class 1 RNRs require two separate proteins for activity. The alpha subunit, which is a dimer, so we call it alpha 2. We have one monomer here and another monomer here. This is the protein that binds nucleotides. Okay, so we have the active site here, which is where the substrates bind, and then we have at the dimer interface one allosteric site known as a specificity site, and it can bind a DNTP, so a downstream product. And this controls the identity of the substrate that's reduced at the active site. The other protein that's necessary is the beta subunit. So it's also a dimer, so beta 2. And what's important is that this subunit contains the cofactor, a radical cofactor that ignites the chemistry. And so for function of this enzyme, these proteins need to come together and at least form a transient complex to engage in this long distance intersubunit radical transfer that's necessary to initiate catalysis at the site. And so the way activity regulation works in class 1 RNRs is through this use of this third domain that I've colored in yellow, the third site. Okay, and this is called the activity regulating site. It's part of this sort of conserved fold known as the ATP cone domain. Okay, this is part of the same chain as this monomer, but it forms this 4 helical bundle, and it has evolved to be able to bind ATP or DATP. And through many studies, it's clear now that when DATP binds this motif, it promotes protein-protein interactions to form these really interesting, unusual ring-like structures. And the common sort of mechanism is that these ring-like structures prevent a productive conformation of the alpha and beta subunits for radical transfer. And so this is how you would downregulate the activity of a radical enzyme. And I'm going to draw your attention to this particular one right here from Pseudomonas aeruginosa, because it gives us some insight into how, through evolution, we could gain and lose allostery in the RNR family. And this is a cartoon representation of the first part of the sequence of the Pseudomonas aeruginosa alpha subunit. And what these authors found was that, interestingly, this protein has 2 copies of the ATP cone motif. The older copy, the one that's attached to the catalytic domain, was probably functional at some time, but it lost all of the residues needed for ligand binding, probably through sequence degeneration. This is, of course, all a hypothesis. And at some later time, it needed activity regulation, and instead of re-evolving those ligand binding residues, it just attached a new functional copy at the N-terminus. Okay, so what's really cool about this mechanism is that it tells us that one way to gain allosteric function is by attaching a conserved regulatory motif. And there are many examples like this in nature. Okay, but of course I'm going to tell you that that's not the whole story. And for that, I have to go back to the family tree. So now I've colored the class 1s into 2 different groups. This clade belongs to the eukaryotic class 1 RNRs, and the remainder are the class 1 RNRs for bacteria and viruses. Okay, and here are the 3 structures that I already showed you, the ring-like structures. They all have ATP cones. They all form rings when they're inhibited. Okay, and in fact, the ATP cone motif is very prevalent throughout the RNR family. You can find them everywhere. But one part of the family that definitely is not is this clade right here, the class 1Bs. So the class 1B RNR was really interesting to us because it evolved out of the bacterial class 1 RNRs, probably as through a gene duplication event as a way for bacteria to develop a secondary RNR. And actually bacteria are very well known for having multiple copies of the same enzyme. But through that duplication event, it actually lost part of the sequence. And it lost about half of the ATP cone motif. So here, shown here, is a prototypic RNR with an ATP cone from the E. coli RNR. And all class 1Bs actually are missing the first 50 residues out of this 100 residue motif. and it's missing all of the residues important for ligand binding. So when it was first discovered several decades ago, it was considered a subset of RNRs that has no activity regulation. So then why would we study this, a subset? It's because the class 1b RNRs are used almost exclusively by bacteria. And that includes common human pathogens, such as Bacillus anthracis, which is anthrax, tuberculosis, pneumonia, and so forth. And so this was the reason our collaborators in Joanne Stubby's group at MIT was studying the class 1b RNRs, as well as all the other RNRs. But before I get back into that story, first I want to tell you, how would we know if an RNR has this activity regulation? And so this is data from Mac Parker, who was a graduate student in the Stubby lab. And he showed that in the presence of ATP, this enzyme is able, it stimulates activity of the enzyme, so SA specific activity. In the presence of DATP, first we stimulate the activity, okay? And this is expected for class 1 RNR because I showed you that the alpha subunit is a dimer and DATP is a specificity effector. It can bind up that interface and stabilize it. But the key signature of activity regulation is that at higher concentrations of DATP, there's a second site for DATP, and now it acts as a potent inhibitor, okay? So these are the hallmarks of activity regulation with an ATP cone. The catch was that this was a class 1b RNR, and that was totally unexpected, and the first example of such a class 1b RNR. And this was the enzyme from Bacillus subtilis, okay? So this is where Will took over and did a lot of structural studies. And in my lab, we like to use small angle X-ray scattering, or SAX, quite a bit. And one flavor of SAX that we like to do is chromatography coupled SAX. And we're known for bringing our own columns to the synchrotron. But one type of column we like to bring is a high resolution anion exchange column. So in this technique, proteins will elute off the column. They're separated by charge. And as they elute, they flow directly into the X-ray path, and we're collecting data the whole time. So we get many, many scattering images from which we can get a large data set that looks like this. It's a three-dimensional data set that has often hundreds to maybe 1,000 scattering curves. Now, in the Ando lab, when we see data like this, we see a matrix. And what we want to do is do math on it. Specifically, we want to do matrix factorization in order to pull out the scattering curves of the protein components. Anion exchange SAX is a little challenging, and so we had to develop a new technique called Regals. It's a very Regal technique, and it stands for regularized alternating least squares. And if you want to know more about Regals, please go to Steve Meisberger's talk later today in the Frontiers in SAS session, so 2:35. The important thing is that Regals allowed us to cleanly separate the co-eluting protein species from this changing background scattering in a robust way. And this is where our first discovery came, which is that we were able to identify the two species here. The first species we knew had no ligand bound. This was a result from the Stubby lab. And when there's no nucleotide bound, the alpha subunit is a monomer, okay? The second species we knew co-elutes with nucleotide, and I'll tell you what that nucleotide in a second, but the surprising thing was that Will could tell that this was a new type of dimer. It was not the dimer that we're expecting. It was elongated, and in fact, our collaborators in Amy Bull's group at Penn State had solved a crystal structure of it. Okay, so Ileana Maggiolo did this work, and the conclusion is that we were expecting an S-shaped dimer that looks like this. This is what we had seen in all class 1 RNRs before, where the dimer interface is formed by the specificity site, which I'm now going to call the S site, and this forms the S-shaped dimer. Okay, we were expecting this, but instead discovered a new type of dimer, which the dimer interface is now composed of this truncated ATP cone motif. This is completely unexpected. And it was stabilized by the binding of these nucleotides. And this nucleotide was DAMP, which is deoxyadenosine monophosphate. Now, at this point, there was no known role for DAMP in the RNR literature. This was a nucleotide that we were not expecting. but it was clearly there, and the N-terminus had evolved this new site for it. And it stabilized this new dimer, which we call the I dimer for its shape, the letter I, but also for DAMP being a partial inhibitor. So this was our hint that there was something interesting going on. And so Will went on to do a lot of SAX experiments, and I don't have time to go through all of them, but just to give you an idea of what I mean by matrix factorization, we use all of these mathematical techniques to not only understand what sort of structures are interconverting, but the nature of those transitions. Okay, so I'm going to summarize a lot of SAX data in a few bullet points, and I'm just to sort of describe one part of the mechanism, so the inhibition mechanism. So from SAX, we learned that one molecule of DAMP binds one monomer of alpha, and it produces an I dimer. Okay, and we knew this from singular value decomposition as well as structural analysis. We also knew that DATP can convert both of these species into the same final species. Okay, so monomer as well as this I dimer. And we knew this from singular value decomposition. We also knew from structural analysis that this final state was a filament that looked like a straw, so a hollow cylinder. Therefore, we knew that there must be two binding sites for DATP, and that one of those sites is shared by DAMP. So it's able to kick out DAMP, bind the other site. Furthermore, this final state, this filament, must include the I dimer. So like any good structural biologist, Will made a structural model, which I like to call the packing peanut on a Post-it model. And it's actually a very good way to illustrate our thoughts on this. So Will hypothesized that we can have two molecules of DATP binding the S-dimer interface to stabilize the sort of canonical S-shaped dimer. But we also knew that DAMP can bind the N-terminus, and we had good evidence that DATP can replace it. And so an I dimer interface must also be able to form. And if we alternate these, we should be able to form sort of a non-terminating polymer of alpha subunits, and that it should have a helical nature based on what we knew about the structures. And based on a lot of the SAX data that he had, it was very relatively straightforward for him to solve a cryo-EM structure. So within a few months, we had our first cryo-EM structure. And indeed, it was a helical filament. This is a structure from a 4.8 angstrom resolution map. And just as he had predicted, it was a helix, which was very cool. And if you look down this, this helix, it's actually a double helix, so it looks like a straw. And so that was interesting. If we look closely at the map, just as we had predicted, DATP was bound at both the S-dimer site and then the I dimer sites. So that was very cool because we had sort of predicted this through SAX, and we were able to verify it through structure determination from cryo-EM. Now, Will went on to do the same strategy, did a lot of SAX experiments with different nucleotides to be able to develop this very detailed allosteric model. And from all of this, he was able to very rapidly solve many structures using higher resolution techniques. I'm not going to talk about all of them in detail, but he was able to get multiple crystal structures, 2 cryo-EM structures, and a very clean scattering profile that told us about the active state of the enzyme. I'm going to focus your attention on the crystal structures because this told us about the evolution of allostery. So here is the crystal structure, or one of the crystal structures. And what we found was that there are 3 allosteric sites, okay? And if we take a closer look, this is an S-shaped dimer, this S-dimer. The dimer interface is stabilized by the specificity site. So the S site is one that is highly conserved throughout the RNR family, okay? And it's evolved actually to distinguish the different DNTPs that can bind there. So it can accommodate all 4 DNTPs. And the identity of that determines the identity of the substrate. And if you want to know about that mechanism in the class 1 RNRs, I highly recommend that you read this paper by Christina Zimani. Okay, but then there are 2 other allosteric sites that evolved in this truncated N-terminus of B subtilis RNR. And the first one, the I site, which is where we initially found DAMP bound, turned out to be the on-off switch for this enzyme. And what was very cool is that we were able to find that this site had evolved to become adenine specific. So there are many residues here that prefer the binding of an adenine nucleotide. Additionally, there's an arginine that can interact with the 2' hydroxyl group, and this was what recognizes whether it's a ribonucleotide or a deoxyribonucleotide that binds at this site. And depending on the identity, that residue is part of sort of an allosteric network that communicates with the I-dimer interface and determines whether it forms this I-dimer or it dissociates that I-dimer interface. Now, interestingly, there's no, from our structural data, there was no reason to believe that there was any discrimination for the number of phosphates on the nucleotide. And that's why we were able to find DAMP initially bound at this site. Okay, so the I site evolved in this N-terminus as a convergent form of activity regulation, mimicking RNRs that have the ATP co-motif. There was a third site that we found, which we call the M site for monomerization. And unlike the I site, it's less specific. It's in fact the least specific out of the 3 sites. It's purine, it prefers purines and prefers guanine nucleotides over adenine nucleotides. And what is very interesting about the M site is that it's only involved in activation. That's because when any nucleotide is bound there, it favors the dissociation of the I-dimer interface because it's exactly where the I-dimer interface has to form. So in my lab, we like to sort of think about these 3 sites as maybe different stages in evolution based on how much specificity is there. Of course, this is all, hypothesis, but some way to think about evolution of allostery. And through those allosteric sites, B subtilis RNR had developed a really elegant form of convergent allostery, where DATP always favors this formation of a filament. So here's a cryo-EM micrograph. And what we found was that when the beta subunit is there, it actually dissociates the double helical alpha filament, and it binds down that central hole of that straw. And interestingly, it binds in such a way that the repeating units are alpha 2, beta 2, okay? And this is the oligomerization state that is needed for activity. And ATP, which is the activator, is actually able to break apart the filament exactly at those I-dimer interfaces to siphon off these alpha 2, beta 2 units. And once they're on their own in solution, it's able to access the full range of motion needed for activity. So DATP sequesters and ATP acts as a siphon. Okay, so with that, I'm going to conclude the first part of my talk, and I want to summarize this part with a couple of points on what do we learn about allostery. So again, here's a cartoon of the prototypic RNR that has the ATP cone sequence. And I'm going to tell you that the majority of the RNRs that have activity regulation have this motif, okay? So it's very unexpected that a member of the class 1B RNR had evolved a convergent form of allostery. And this told us that allostery can emerge without copy pasting an existing conserved motif. And furthermore, I told you already that the residues involved in ligand binding were not transferred to in this truncated sequence, and they weren't even localized within this N-terminus for the B subtilis RNR. And I think this is particularly interesting because what this is saying is that, of course, within a protein, there's no discrimination of this part is a regulatory part and this part is not. And for me, this is some kind of evidence that there is an allosteric network that can emerge from the protein scaffold of a protein structure. That there is no reason that allostery should evolve in just a designated region. Okay, so with that, I hope I planted a seed in your mind about the idea of allosteric networks. And for that to emerge, macromolecules and proteins in particular have to be able to undergo correlated motions. And so that leads into the second half of my talk. This is the work primarily of Steve Meisberger, postdoc, whose talk you should also go to later today. I've already told you multiple times. And also David Case, our collaborator, who did a sabbatical in my lab for a year. And if you're interested in learning about the theory of diffuse scattering, I recommend that you read this 60-page chemical reviews article. It was a lot of work to write this review article, so I also do want to acknowledge the co-authors on that with their photos. But the story I'm going to tell you about now is primarily from this paper from earlier today. Okay, sorry, earlier this year. Okay, so before I talk about diffuse scattering, let's go back to the basics and define what is a structure. I think everybody at the ACA likes structures. Let's remind ourselves what a structure is. And I'm going to do that by telling you a little bit about this crystal structure, one of my favorite crystal structures. This was a vitamin B12 dependent complex that's involved in microbial carbon fixation. And it was solved by Yan Kung, who is a graduate student in Kathy Drennan's lab. Now, the goal of crystallography and actually any high resolution structure determination technique is to be able to determine the coordinates of every atom in your structure, right? And so if you download a PDB file, so this is from the PDB file of this particular structure, you'll find that information. So here's a nitrogen on lysine 23, which is found here in the structure. And you can see that with this atom, there are 5 numbers associated with it. The first 3 are the coordinates, X, Y, Z. So this is actually the average position of this atom. Now there are 2 other numbers that tell us about the probability that the atom is actually at the average position, okay? The first one is occupancy, which we usually set to 1 because we often can assume that an atom is covalently bound to another one. And the second one is the B factor, okay, which formally is the mean squared displacement U from the average position. And there is a B factor whenever there is variability in the atomic position from unit cell to unit cell, okay? And so it's not expected that every unit cell is identical to each other. And oftentimes we describe that through the B factor. We also often use the B factor as a measure of atomic motion. And in the case of this crystal structure, it made sense to think of it that way. So if we were to color the crystal structure in terms of the B factor, you could see that these domains, the B12 domains, really pop out. They have high B factors. And it made sense because this part of the protein made no lattice contacts. And in fact, it's holding on to this cofactor for chemistry. And we had spectroscopic evidence to show that it was in fact moving in the crystal and allowing turnover to occur within the crystal. Okay, so now that I've reviewed what a structure is, let's talk about where this information comes from. Many of you at the ACA know that the information for crystallography comes from Bragg diffraction. We measure the intensities of these Bragg reflections, and by measurement of many, many observations of these reflections, we're able to refine a structural model that has many parameters. Okay, and what we learn in textbooks is that the scattering from a crystal structure is described by the structure factor, okay, which is just a function of the electron density, the Fourier transform. And this is why it's a structural technique. We're probing the distribution of electrons in our sample. What we measure is the intensity, which is the square of the structure factor. Okay, but what we don't often think about is the fact that what we're actually measuring is the average. You could think of this as a time average, because atomic positions do vary from unit cell to unit cell. They fluctuate, and sometimes there's static disorder. And so what is the consequence of the fact that we're actually measuring the average? That is that there are actually 2 parts of the signal, and we're only using one in crystallography. So the Bragg signal is the lattice term part of this, the total scattering. And we know from crystallography very well that this is, we're probing the average electron density of the unit cell. So we're taking the average of the structure factor before the square. And this, in this very simplified form, you can tell that the 2 is in the outside here and on the inside here. These 2 are not the same. So whenever there's variation in atomic position, these 2 are not the same, and that gives rise to a different signal, which is the difference between the 2. And this is called diffuse scattering. And diffuse scattering is a continuous function. Okay, so what does crystallography give us? We can tell from this very easily that we're probing the average electron density of the unit cell, which is why we were able to get the atomic coordinates. And from the decay in the Bragg intensities that come about from any disorder, we can also get the B factors and get a sense of the atomic displacements. Okay, but that decay in the Bragg intensities gives rise to the diffuse scattering signal. So we're not losing photons, it's just being diverted to this different signal. And why do we care about the diffuse scattering? It's because this is the only way we can get information about how the atomic displacements are correlated. And to illustrate that, let me just give you three, these sort of simple models. In all three cases, we have identical helices that are moving, and they're being displaced by the same amount, but they're undergoing different types of correlated motions. Now, by Bragg diffraction, we can't distinguish these three cases. The electron density will be blurry by the same amount. The B factors will be the same. However, these different motions will give rise to distinct fingerprints within the diffuse scattering. Okay, so the take-home message is that from Bragg data alone, we cannot get information about correlated motions, and this is really ultimately what we want to get to understand in structural biology if we want to understand the relationship between protein dynamics and function. So the problem is it's been very, very difficult to interpret diffuse scattering from protein crystals. And X-ray diffraction has been around for a very long time. And it was first demonstrated by very famous son and father pair, William Bragg and William Bragg. And it's been known for decades that diffuse scattering contains this information and has the potential to tell us about protein dynamics, but it has not yet been realized. So I'm not going to be able to tell you about the history of the macromolecular diffuse scattering field. There are many, many important milestones, but the bottleneck until recently was measurement. We did not have accurate enough data to be able to interpret it. And so our approach was to make use of the fact that pixel array detectors had become prevalent at macromolecular crystallography beamlines. This was a game changer. And here again is Steve Meisberger leaning against the $3 million detector, as its cover on it, so it was not damaged. And then we used our knowledge of scattering to tackle this problem. And to summarize a lot of things, we had to come up with a new experimental approach to be able to collect low dose exposures that are finely sampled on crystals at room temperature. So we collected many partial data sets and then merged those. The key feature of this data collection procedure is that we used a pixel array detector where the photon statistics are well known. And we also collected, measured the background, just like in a SACS experiment. And we also made sure that the data had high redundancies. We're measuring the same, making the same observations multiple times, just like we do in crystallography. And using this type of data set, Steve built a new software pipeline from scratch, which we're currently called MDXlib. If you want to know more about it, please read this paper. I can't tell you all the details, but here are some of the key features. First of all, utilizing the fact that we know that the detector should have certain statistics, we're able to develop this very sensitive filter that gives us an integration mask. Utilizing the high data redundancy, Steve developed a way to do a global refinement of a scaling model that corrects for many of the experimental artifacts that come from measuring scattering from crystals. And also importantly, he developed a way to put the intensities on an absolute scale of electron units. And this is very clever. And this is very important because for one, it allowed us to be able to subtract the inelastic contribution to the scattering, which contains no structural information. But also by putting intensities on an absolute scale, we force the modeling to be more rigorous because it places a stringent requirement that the model has to also explain the intensities absolutely. And then through the refinement of these processes, we ultimately are able to place the diffuse scattering onto a fine grid in three-dimensional reciprocal space to ultimately get a data set that looks like this. We like to call this the Death Star, but you can see that it's very finely detailed, and here's like a slice through into this Death Star. Okay, so this Death Star was collected from diffraction from triclinic lysozyme. So we have data on many crystals, but we started by analyzing data from this particular crystal for a number of reasons. One was that it had extremely low mosaicity. So the angular spread of the Bragg peak was very small. It was only limited by the instrument, the beamline hardware. And that allowed us to distinguish diffuse scattering and the Bragg diffraction very well. The other reason why it was important to start with this was because there's only one protein per unit cell, and the unit cell dimensions are very small, which means that the Bragg peaks are well separated, and we're able to sample very finely in between them. And so this map actually is just the diffuse signal. You can see that's very detailed. This final map has 50 million unique observations. Okay. Now with this fine detail, we were able to see lots of interesting things about the diffuse scattering. First of all, I'm going to ignore these isotropic contributions, but we were able to separate them very cleanly. And we were able to describe the diffuse scattering as two types of signals. There was a sort of smeary, sort of slowly varying component, which we called cloudy diffuse scattering. But there was also very clear, bright scattering near the Bragg peaks. So there are no Bragg peaks in this image right here. This is diffuse scattering, and we call these halos. The presence of halos was very surprising, but we were able to see it because of the fine detail. And because of the fine detail, Steve was also able to analyze this further and show that there's a distinct power law decay away from the Bragg peaks that was indicative of long-range correlated motions in the protein crystal. And in fact, it was strongly suggestive of the fact that we have what are known as acoustic phonons. So these are lattice vibrations. And this was very surprising because, well, it had been suspected and proposed that acoustic phonons may occur in protein crystals, but there was no strong evidence prior to this point because it had been so difficult to measure such diffuse scattering on a fine grid. But with Steve's data set, we could see, this data point that is closest to the Bragg peak told us that at the very least, there are unicells that are able to communicate. They're correlated over a distance of 300 angstroms, which corresponds to about 10 unit cells. So really long distance communication. And we got sort of additional support for this from MD. This was the work of Dave Case, where he did an all atom simulation of, he actually did many of them, and I'm just going to show you a subset of simulating the triclinic lysozyme crystal. So the very first one he did, he simulated a single unicell and then applied periodic boundary conditions. And then we calculated the diffuse scattering from that sort of converged simulation, and it looks like this. You can see it's very gradually changing. This looks like cloudy diffuse scattering. However, when we increase the simulation size, right, so now we have 27 unit cells that are in this periodic boundary condition, we start seeing these brighter features. And he went on to do really giant simulations, and finally up to 343 unit cells, the supercell simulation. And now you can really see that there are strong features where you would expect Bragg peaks. And so this also supported the idea that if you allow the simulation to occur over long distances, you will start capturing this sort of long-range correlations between proteins in different unit cells. But we really wanted to understand the physics that gives rise to this. And so Steve went on to do his own simulations using elastic network modeling. So the first thing he did was he made a vibrational model of lattice dynamics. And for that, he treated each protein as a rigid body and the lattice contacts as springs. So this is a vibrational model. And then he applied periodic boundary conditions. And then at every sort of stage of the refinement, he calculated the diffuse scattering and then fit it against the halo scattering near the Bragg peaks. The really cool thing is that he only chose a very small subset of the halos, just 1%. So the working data set was 1%. But here's what he saw. So at the end of refinement, so first of all, here's a slice through of the diffuse map, the experimental diffuse map. You can see all the halos, and the halos that are in blue boxes, those are the ones that were actually fit in the simulation. And then here's a refined lattice model that Steve did, and you could see that there is very clear agreement between the two. And we can sort of zoom in further to make this point clear. So in this zoom in, there are only 3 halos that were actually used in the refinement. But you can see that this model was able to describe the diffuse scattering throughout reciprocal space. So that was very cool. The part that was very surprising was that the halo scattering actually extends really far into the Brillouin zone, and it creates this sort of cloudy pattern. So what we had been attributing as entirely cloudy versus halo, it seemed like the halo was actually contributing quite a bit to this cloudy pattern. And so that was surprising. So at this point, Steve decided to take a step back and wanted to understand how much of the diffuse scattering was explained by these long-range lattice dynamics. And he did this by looking at the B factors that I told you about earlier, which is shown here. So these are the B factors from the refinement of Bragg data, so from getting a crystal structure shown per residue. And then he calculated the B factors from his lattice model. And these are the expected B factors from that. And you can see that it actually accounts for a lot of the motions of the atoms, but there is a difference. Okay, and that accounted for difference. The question was, are these due to internal dynamics of proteins? So Steve went on to do another simulation, another elastic network model simulation, but now he had springs within the protein. And I'm not going to go into the details, but the key features are that he did something very clever to make sure that the data to parameter ratio was still high. Okay, right here. And instead of fitting the diffuse scattering, he fit against the atomic displacement parameters. Okay, so this is, you can think of it as expanded form of the B factors. The diffraction data was sufficiently of high resolution that we were able to refine the full set of ADPs. Okay, so what do we see? I'm going to first tell you that, well, I already told you that a lot of the diffuse intensities were already accounted for by the long-range correlated motions arising from lattice dynamics. So it made no sense to try to look at the residual intensities. Instead, we had to sort of do an operation to the intensity so that we can sort of see the contribution of internal dynamics, and a very useful operation is the Fourier transform. Okay. And so this is the diffuse Patterson, the 3D delta PDF, and we're just going to zoom into close to the origin to look at the shortest length scales. Okay, so the circle represents 10 angstroms. And as a reminder, diffuse Patterson is the autocorrelation of electron density fluctuations. A vector starting from the origin tells you about the correlations of a pair distance in the crystal. So here is the diffuse Patterson for the experimental diffuse map. You can see that there are lots of positives and negatives, sort of large amplitudes near the origin, and then it dies off, but it extends quite a bit. And this is in line with the fact that we saw long-range correlated motions, right? So 10 angstroms is roughly the size of the lysozyme molecule, but it's extending past that. And if we were to simulate the diffuse Patterson from just the lattice dynamics model that Steve did, we could see, in fact, yes, there's a lot of agreement, especially going out to past this 10 angstrom circle. But there are correlations that are not accounted for closest to the origin. Okay, and how about the internal dynamics model? This gives rise to this pattern right here. And you can see that it is very concentrated within the circle, as we expect. These are the correlations within the protein. If we add the two now, this was very exciting. We were able to get very close agreement to the experiment. So the lattice dynamics plus the very hard to detect, but is actually there, internal dynamics of the protein gives rise to the diffuse scattering. And I'm going to try to wrap up quickly. What sort of internal dynamics were we seeing in that refined model? Here are the two lowest frequency normal modes with amplitudes greatly exaggerated. But we were able to see that hinge bending motions do contribute to the diffuse scattering and also agree with the B factors. And there's actually much more to the story, so I urge you to read our paper. But this was the closest agreement that we had gotten to explain diffuse scattering in the history of macromolecular diffuse scattering. So there are many, many important milestones, but I'll just like to wrap up this part of my talk by saying that seminal studies were done a long time ago, and we are now finally able to explain most of the diffuse scattering. And this is important because crystallography has only used the Bragg diffraction so far, but that only is about half of the photons that are scattered by a crystal. And the diffuse scattering we showed tells us about these long-range correlated motions, which are have significance in terms of protein-protein interactions, as well as it's also sensitive to the internal dynamics of proteins. And then we hope in the future that we can combine these two data sets to be able to get a fuller picture of protein structure and dynamics. And with that, I'm going to wrap up my talk. I have many people to thank, including many collaborators. These are just the collaborators who are on the papers that were shown throughout this talk. If we run out of time, please feel free to email me. I've starred all of the co-authors on the papers that I've shown here. I want to thank our funding sources. We've done all of our scattering work at Chess. Thank you for your time. And I'll also just thank my group once more because they've not only worked with me very hard on research, but also on other projects, one of them being the ACA meeting itself. So many of them contributed to this meeting, and I'm happy to take questions.
Nozomi, this is Brian Toby. We already have 4 questions waiting for you in the Q&A. I can read them to you, or you can open it yourself as you choose. I think we're up to 5 now.
I'll have you read them.
Okay. The first one was Stephen Kelly asked, in the chromatography SAX experiment, roughly what volume element of elutant is irradiated? Does it work better if you irradiate as much as possible to get a good signal, or if you use a highly focused beam to irradiate narrow slices and possibly improve the resolution between chromatography peaks?
Okay, yes, that's a great question. I'm going to recommend that you go to the Frontiers in SAS session to learn more about this, but actually having higher flux doesn't help. Radiation damage will become a problem, and one way to get around this is using a co-flow setup that APS is doing, I think out of necessity, but you will have radiation damage even if you're flowing the sample very quickly. It will, the aggregates will stick to the windows, and that could be a problem. The irradiated volume is probably, a typical amount, like I would say like a few microliters at most, but the sample has to flow by very quickly. You can change the column to increase the resolution, but there are many things that you can't change, which is the sort of interconversion rates of different species, so that even if you think you're separating them on a column, they're not fully separated. And this is where using our sort of mathematical approaches will really help.
Okay, next question is, can nucleotide analogs such as PPG, PP bind RNP for regulating activity?
That's a great question. That was asked yesterday as well at Will's talk. So that is a sort of a derivative of GTP that is sort of an alarmone. It's a signal that's used in the stress response of bacteria. There may be other biological roles for it that I don't know about, but based on our experiments, we don't see evidence for it doing anything. And I think if we try to fit it in a crystal structure, we'll notice that it actually won't fit. The phosphates on either side of the nucleotide will be a problem. Yeah.
Okay. Let me just ask people who have been typing in questions into the chat box to please put them into the Q&A, because the Q&A is recorded, and we hopefully can get back to questions if we don't have time for all of them. We have one anonymous attendee who said, excellent agreement to the halos with the initial lattice model. How well did it agree before the fit?
Before the fit. So I actually can't remember if I've seen a simulated diffuse scattering from before the fit. That's a great question, and I don't know the answer. I don't think very well, but since I don't know, please come talk to me and Steve later.
Okay. Let me just point out that it's now time for the other sessions to begin. Nozomi, would you be willing to stick around for a little bit longer and answer questions for people who want to stay here? Then that's great.
Yes.
Okay.
Yes.
For those who would like to continue, and I point out that everybody who's at least not anonymous, we have your membership information in the membership database, so there's a possibility of getting back to you as well. All right, so next question is from James Holden, who will be giving a talk himself. I forget which day, is the Ronley Award winner. Could these lattice dynamics be actually lattice statistics, such as defect structures creating long-range distortions that do not change with time? So static versus dynamic disorder.
Yeah, that's also a really great question. And I think James is asking this because that power law decay that I showed you, which is characteristic of acoustic phonons, is also true for static disorder as well. There are a couple of reasons why we think it's a dynamic disorder. And other than the fact that our simulations seem to support this. But in terms of other biophysical methods, there are measurements of the velocity of sound through a protein crystal that would suggest that acoustic phonons are supported in a protein crystal. So that's probably our best bet right now. And in fact, actually, if you look at our paper, Steve calculated the expected dispersion relations from his lattice dynamics model and was able to get sound velocities that are very close to what has been measured.
Okay. Our next question is, okay, how much accuracy can you achieve through the diffuse scattering compared to only using Bragg reflections in terms of standard uncertainty?
Is this for a structural model, I guess?
It sounds that way.
I think Bragg diffraction does very well in terms of getting high precision structural models. It's only when we try to interpret the disorder in a crystal that diffuse scattering really will start to matter. So especially in cases where you want to start making assumptions about certain groups of atoms moving together or as a group, that kind of refinement allows you to reduce the number of parameters that are fit your Bragg data, but that also assumes that there are certain motions that are happening. So classic example is TLS refinement. And this is where having diffuse scattering will improve the accuracy of the model or at least validate it. But I think in terms of statistics, without knowing any knowledge of diffuse scattering, you can still do very well with common crystallographic refinement techniques. It's really more of whether you want to believe in the sort of implied physics of those models.
Okay. Alan Orville from the Diamond Light Source says, great talk, thank you. Eventually mosaic spread will obscure diffuse scattering. How large a mosaic spread can diffuse scattering before diffuse scattering is hard to measure accurately. He has a later question, which I'll put out as well, which is, I anticipate that we will see diffuse scattering as a function of ligand binding or catalysis. Have you tried this in your systems?
Yeah, so for the first question, yes, mosaicity will be limiting. Unit cell size will be limiting. We're trying to explore those limits right now. We have many data sets. So hopefully we'll have an answer soon. In terms of people who are in charge of making hardware for beamlines, please consider the possibility of making sure that we are able to do these measurements. So that means having very low beam divergences and the ability to have very narrow energy bandwidths help. In terms of the second question, which I'm trying to remember now. Can you remember, remind me what the second
Sure.
question was?
Okay, let's see. I anticipate that we will see diffuse scattering as a function of ligand binding and or catalysis. Have you tried this in your systems?
Yes, we are analyzing data right now, and it is going to be very subtle. Like I told you, the internal dynamics contribute to a very small amount of the total diffuse intensity signal because the long range motions do dominate. But we do have data, and we're looking into it right now.
Sounds good. All right. We can stop at this point, but I'll give you the last few questions that were asked before the talk ended. Let's see. So can you tell us anything about how disordered solvent does or does not contribute to diffuse scattering?
Yes, I didn't talk about it because we subtracted it from our data. That contributes to an isotropic diffuse scattering ring that I think many of you are familiar with, around 3 angstrom. That's mostly solvent. There's some protein contributions to that as well, but that didn't contain the information that we're interested in. So we subtracted that from our diffuse maps that I showed you later.
Okay. So David Rose says, terrific talk, congratulations. Are there any other examples of the B subtils like mechanism that are known in other RNRs?
No, this was the first one. And now we're exploring this further. What I really like about the class 1B RNRs is that because of that truncation, this subset of the family has a very sort of clean, sort of very well-defined sequence length. And so when I look at the sequences of class 1B RNRs, to me, it looks like a matrix. And I'm very excited about looking at how allostery emerges within this subfamily. And that is actually ongoing work in the lab.
Okay. 2 more questions, actually 3, but have you compared X-ray diffusions from unfrozen and frozen crystals to see how freezing crystals influences protein motions?
Yes, actually, we started our work by looking at cryo-cooled crystal diffraction data. And the first thing I can tell you is that there is diffuse scattering there. The reason why we went to room temperature, though, is because the act of cryocooling a crystal can also introduce disorder. And so we can strain the lattice that will create some kind of static disorder, and that will give rise to diffuse scattering. And we really did not want to worry about that. And so we moved away from that. But it is a question that many people ask, whether temperature has an effect on the dynamics. And we would like to eventually do that comparison. I don't know if going all the way to cryo is the best way to do it initially.
Sounds like to me like it's the opposite of what you want to do. Okay, Amy Sargent asked, thanks for the excellent talk. Thanks for the great balance between background information and detailed research discussion. So that's actually not a question. But anyway, Branton Campbell in the chat box said, could you say something about your workflow for reconstructing the 50 million voxel diffuse scattering data from the raw scattering images? I'm sure you'd love to say a lot about that, but we don't have time on that.
I'm going to suggest that you go to this Frontiers in SAS session, and then you can mingle with Steve at 3 o'clock. The software pipeline was, it was a massive project. I think thousands and thousands of lines of code went into it, and I won't do it justice in just a few seconds. So I urge you to come chat with Steve.
Okay. All right. Thomas Profen and I bemoan the lack of a clap button, but I really would like to thank you for a wonderful talk, and I'd like to thank you also, you and Carla, for a wonderful meeting.
I would also like to thank Carla, and thank you everybody for joining me this morning.